Hello everyone, my name is Farrah Shalima Mohammed. I'm a sixth-year PhD student in biomedical engineering at the Yale lab at Yale University. I wish I could be with you all in person, but I'm so grateful for this opportunity to speak with you and share my passion for nanotechnology, tissue engineering, and finding treatments for traumatic brain injury. Today I'll discuss my review that I published in 2022 entitled Nanoparticle-based drug delivery for the treatment of TBI, but the QR code is actually located on the top left-hand corner of every slide today, and if you don't have access to this particular journal, no problem, I'm more than happy to send it to you, so shoot me an email. Towards the end, I'll discuss a bit about my personal research in the field, and in the end I'll have some links so that we can stay connected. An estimated 69 million individuals suffer from traumatic brain injuries each year. While the incidence of TBI is highest in regions with higher quality data, like North America and Europe, the Southeast Asian and Western Pacific regions experience the overall greatest burden of disease. While traumatic brain injuries affect the breadth of society, the marginalized are the most affected, including victims of assault and people experiencing houselessness. Interestingly, 5 cosmonauts who spent over 169 days on the International Space Station showed heightened levels of neurofilament light chain, GFAP, and amyloid beta 40, indicative of brain injury and neurodegeneration. So as you can see, not only globally and across a certain level of society, but all levels of society, age group, profession, experience some level of risk to traumatic brain injury. So greatly, there's a need for treatment. Traumatic brain injuries, or TBIs, constitute both closed head injuries, like hitting your head on the floor, where there's a greater chance of blunt trauma, rapid acceleration and deceleration forces experienced by the brain, and less commonly penetrating injuries, for example, where an object perforates the skull, breaches the meninges, and injures the brain. You can imagine this like a gunshot wound. The pathophysiology of TBI is biphasic, so the primary injury is not treatable. Here you can see maybe more preventative measures being put in place, like wearing a helmet or having better tackling practices, but this primary injury is the direct consequence of the mechanophysical forces responsible for the insult. The secondary injury now is the phase that represents the only window for intervention and is the body's attempt to limit the extent of damage and restore the brain's structural and functional integrity. Here, for example, you see we call it coup contre coup, which is in the beginning you have this forward injury or the first injury, right? And then after the reverberation, you have other areas of the brain that are affected as a result of the recoil. And so the physical, cellular, and biochemical processes that occur during the primary injury progress into the secondary injury mechanisms on a time scale of about seconds to years even, and this results in cerebral swelling, herniation, and neurodegeneration, which can lead to cognitive and motor impairment, and in some cases, even death. So the blood-brain barrier is actually what we're super interested in, and this is a selectively permeable microvasculature that shields the brain from toxic substances in the blood, provides nutrients to the brain, and relinquishes harmful substances in the brain to the bloodstream. At the moment of impact, the BBB is often compromised, so here we have triggered a downregulation of the platelet-derived growth factor beta signaling pathway, impaired parasite endothelium interactions, and this all results in parasite loss near the BBB, further compromising the BBB's integrity. This dysfunction of the BBB dynamics is followed by an increased permeability to water, marked by a significant increase in aquaporin 4 expression and possibly edema. Also contributing to this brain swelling is the ruptured BBB, and this is the mechanical rupturing, right? And this encourages vasogenic edema, where protein-rich fluid from the circulating blood enters the brain interstitial fluid. Cytotoxic edema can also form where there's a dysregulation of homeostatic ions, channels, and pumps that disrupt the ionic gradient, leading to intracellular swelling of astrocytes and glial cells. Both cytotoxic and vasogenic edema can elevate the intracranial pressure, and this worsens functional outcome, or is correlated to. So the severity of a TBI is usually assessed using the Glasgow Coma Scale, but this measurement is quite weakly correlated with survival and functional outcome, and so other imaging techniques are employed, such as CT or MRI. The clinical management to reduce secondary damage and improve the prognosis of motor and cognitive functioning remains a challenge, since there's no pharmacological agents that have succeeded phase 3 clinical trials. So for example, like progesterone has gone to later phases but failed to improve outcomes, notably due to poor drug delivery efficiency and inconsistent distribution of drug volume, and also there's some off-target effects. So because of this, there's a need to find some approved treatments. Here I'd like to expand into the deleterious processes a bit further. So after the BBB is compromised, these damage-associated molecular patterns, or DAMPs, are released from injured cells and are recognized by pattern recognition receptors. So in response to the DAMP signals, an inflammatory response is elicited by leukocytes, microglia, and astrocytes, which release pro-inflammatory molecules in a positive feedback loop. Prolonged inflammation promotes astrogliosis, resulting in a scar-like formation around the injury perimeter and a reversal of glutamate uptake mechanisms, which can contribute to a persistent activation of NMDA, AMPA, and voltage-gated ion channels, resulting in a lethal influx of calcium and subsequent neuronal excitotoxicity. Macrophages, microglia, and other cell types also respond to DAMPs by activating pro-inflammatory pathways such as the NFKB inflammatory pathway, which can contribute to edema and neuronal apoptosis. The pro-inflammatory mechanisms and molecules described, along with the glutamate accumulation from the shared neurons, like due to the injury, impair reuptake mechanisms, and neighboring glutamate-induced aggravated release from presynaptic terminals, exacerbate excitotoxic mechanism, reduce ATP production, which eventually leads to mitochondrial dysfunction, increase oxidative stress, and eventually lead to cell death. And this cell death triggers further release of DAMPs, and as you can see, this process starts all over again. To investigate traumatic brain injury treatments and strategies, researchers are faced with the challenge of choosing an appropriate injury model to investigate the heterogeneous nature of TBIs in vivo. Broadly, animal models of TBI are classified as either mainly focal, diffuse, or mixed injury models. Ideally, when designing an experiment, the model should be tunable, commensurate with the desired injury severity, quantifiable, reproducible, and have some clinically relevant correlation. As you can see with all of the methods that I've described here, there are advantages to some and disadvantages to some. So some common methods to mimic a focal brain injury, for example, include a weight drop model. The open skull control cortical impact model is also pretty common, but for example, like I said, that there is advantages and disadvantages. That model requires a craniectomy, and that in itself is an injury. So with everything, you have to think about design, right? And so here I use this model actually, but all of my shams include a craniectomy without an injury. You also have the penetrating ballistic injury model. Diffuse injuries are commonly modeled using blast wave models, and mixed injury models can be used by weight drop injury, acceleration deceleration models, and fluid percussion injuries. Some techniques are more involved than others. For example, if you have a ballistic-like model or a blast model, you may require to hire some pyrotechnic experts. Traumatic brain injuries present on a spectrum of focal to diffuse types, and therapeutics must be able to penetrate both the intact and compromised BBB, not only to address the injury continuum, but also to be used in both acute and chronic therapies. Nanoparticle technology is best fit to meet these demands, as their modifiable chemistry, size, charge, and customizable functionality harness the potential to foster payload protection and improve therapeutic efficiency, avoiding off-target toxicity and biodegradation. After reading a lot of papers, I can see that the failure of many phase 3 clinical trials is that in order to successfully improve TBI outcomes, these novel pharmacological interventions must efficiently deliver therapeutics to the targeted injury area and be retained in the brain long enough to confer its therapeutic effect while avoiding off-target toxicity. So there are some other criteria I found as well. 1, it should be easy to formulate, stable at clinically relevant temperatures, and easily stored and transported both for acute and long-term treatment. So you can imagine, say on the battlefield, if you need to make some type of clotting agent, right? This nanoparticle needs to be able to be survivable in ambient temperatures and also stored for a while if there's no medical facility nearby. Overall, I'm looking at the different types of research that's been published right now in the nanoparticle space, and they're broken down, though not mutually exclusive, into nanoparticles that confer some immunomodulation, hemostasis, neuroprotection, anti-inflammatory and antioxidative actions, and stem cell therapies. A lot of them are complex as well, so by no means do I think the future is one of these answers. It's probably really a combination of many of these factors, but this is how I'm breaking down the review. So, the first category I'll go over is these immunomodulatory nanoparticles, which are those nanoscale technologies that alter the immune system to improve TBI outcomes. In this field, investigators have focused on designing nanoparticles that mitigate upstream inflammatory players and sequester inflammation both in the vasculature and around the injured parenchyma, sometimes solely through surface modifications, omitting a payload completely. So, for example, you can see the first particle on this list sequesters positive hematogenous monocytes to the spleen, mitigating the inflammatory status of infiltrating cells, and they saw something like a 44% reduction in the lesion volume. Biomimetic nanoparticles, such as the leuconanoparticles listed, can accumulate in the brain through paracellular diffusion and breaks in the BBB. So these provide stealth by evading clearance by the mononuclear phagocyte system, reducing elimination by Kupffer cells in the liver and glomerular filtration in the kidneys, while still maintaining the delivery capabilities of a synthetic nanoparticle. Also in the arsenal to modulate the immune response were these short hairpin RNA-loaded nanodendroplexes, which were shown to attenuate inflammation after repeated TBI and increase this BDNF, the brain-derived neurotrophic factors. So future studies in this field should continue to consider how immunological signaling regulates secondary injuries post-TBI, keeping in mind this timing of this pro versus anti-inflammatory processes. All traumatic brain injuries, most commonly blast injuries, are susceptible to bleeding, and immediate intervention is among one of the most effective solutions to minimizing mortality risk. The only available treatments are pressure dressings and absorbent materials, which are only effective for superficially exposed wounds and are unable to ameliorate internal injuries. To address this, investigators are developing these hemostatic nanoparticles. They're aimed to stop bleeding and slow the progression of inflammatory processes. So commonly functionalized to hemostatic nanoparticles is the synthetic peptide GRGDS, which contains the amino acid sequence RGD, which mimics recognition sites in the interaction between extracellular matrix molecules and cell membrane receptors. When platelets are activated, they expose the glycoprotein through 2B and 2A receptor, and the RGD can block the receptors of these glycoproteins, inhibiting fibrinogen binding and modulating hemostasis through antithrombotic actions. For instance, one study used these 500 nanometers PLGA nanoparticles that had PEG and GRGDS motif conjugated onto them. They were synthesized and loaded with dexamethasone, an anti-inflammatory glucocorticoid, but just the nanoparticles alone were shown to have an elevation in animal survival and significantly mitigated hemorrhaging. In a more recent study, there's a carboxyl functionalized biodegradable polyurethane nanoparticle that delivered spongostan or spongostan, and it's a hemostatic agent that has shown that this PU nanoparticle containing gelatin attenuated brain edema, suppressed expression of M1 biomarkers such as IL-1B, and elevated M2 biomarkers, reduced the activation of inflammatory cells near the implant site, and actually increased BDNF by almost threefold. So here you can see in the first particle I talked about where there is a lowering in GFAP compared to the injured animals, and in the second study that I mentioned, you can see that compared to the saline only, these polyurethane nanoparticles were able to decrease the inflammatory response shown by IBA1 as well as GFAP. While there are other categories of nanoparticles that can confer neuroprotection downstream, those discussed in this particular neuroprotective nanoparticle section contains agents in which their primary function is to elicit a direct neuroprotective action. For instance, many studies have utilized the nanoparticle encapsulation of short interfering or silencing RNA to deliver gene therapies across the BBB to improve neuronal pathology post-TBI, avoiding off-target effects that can be seen with some small molecule drugs. We see evidence in the potential for such nanoparticles in studies like neuron-targeted nanocomplexes TV-RVG nanoparticle, which was actually shown to target injured neurons and deliver siRNA to reduce neuronal apoptosis, or in this tau silencing RNA-loaded nanoparticle, which was shown to reduce tau expression in cultured primary neuronal cells and achieve around a 40 to 50% tau silencing in TBI mice, regardless of being administered within or outside of the window for a physically breached BBB. So That's already ticking one of the criterias I have. Peptides have also been studied in nanoparticle systems to confer neuroprotection, including BDNF, cerebrolysin, glial cell line-derived neurotrophic factor, nerve growth factor, and the list goes on. Including in that is this TAT NR2B 9C, which is a postsynaptic density protein 95 inhibitor thought to block the induction of NMDA-dependent excitotoxicity. In one 2019 study, activatable protein nanoparticles activated to release their payload by proteases, which are actually rich in the injury microenvironment, encapsulated these TAT NR2B9C conjugated with PEG and peptide CAQK, which actually has a high affinity for extracellular matrix at the site of the injury, and they both showed brain penetrability. And then this CCAQKTN activatable nanoparticle demonstrated a higher brain targeting efficiency, reduced injury size, and improved cognitive outcomes. There are other studies using this CAQK and conjugating them to, for instance, silver nanoparticles for targeting chondroitin sulfate proteoglycans, rich scars on mature oligodendrocytes for the injury, and this addresses a major barrier to regeneration. Lastly, there are some natural sources that I think are worth exploring. For example, there are a lot of studies involving curcumin. One study was looking at these orally administered niazomes after transplantation of human neural stem progenitor cells for 10 days, and the combinatorial treatment showed a significant improvement in brain edema after TBI, increased locomotor activity, and decreased astrogliosis and inflammatory factors like this NKKB and TNRA positive cells. Nanocoffee nanoparticles are another example of potential sources of therapies, and the list goes on. As I touched on previously, the pro-inflammatory and oxidative pathways confer deleterious effects that are exacerbated by that positive feedback loop as seen in those promoted by DAMPs in the secondary injury processes. So this category that I created here actually explores those nanoparticles which directly influence an antioxidative or anti-inflammatory effect. As a demonstration of form and function, I'd like to bring your attention to the crystal lattice of cerium oxide, which has multiple valence states that confer its high redox capacity. After a fluid percussion injury, the higher concentration and dosing paradigm of cerium oxide nanoparticles were actually shown to increase neuronal survival, preserve endogenous calcium antioxidative activity, reduce oxidative stress, in part through improving superoxide dismutase activity, and restored reduced glutathione and oxidized glutathione ratios. Interestingly, the shape of the cerium oxide nanoparticles affected its efficiency. Ceria nanorods, which are around 130 nanometers, were shown to exhibit better antioxidative activity and lower cytotoxicity than ceria nanospheres, which were only around 3.5 nanometers. And this is due to nanorods displaying a more high-energy surface with more active sites. More recently, my lab synthesized butylinic amine derived from an antioxidative herb, E. ulmoides, which can penetrate the BBB via cannabinoid receptor 1-mediated transcytosis and release its payload in acidic environments. In this study, we tested it in a stroke model, but the microenvironment of stroke is not that dissimilar of TBI. In our published study, BAM-delivered NA1, which is a peptide designed to protect neurons against NMDA receptor-mediated excitotoxicity, and these nanoparticles increase survival and improve the infarct volume by around 70%. So in this way, you can see that these anti-inflammatory, antioxidative nanoparticles have a great potential for reducing those deleterious effects of the secondary injury response. The last category that I'd like to touch upon is the category that includes those nanoparticle complexes that enhance stem cell integration to repair the injury site, promote tissue regeneration, and improve TBI outcomes. Mesenchymal stem cells are of special interest to cell therapy approaches as their autologous origins evade immune response, display inherent pluripotency, and demonstrate reparative properties at the site of injury. For instance, in an in vitro study, gold nanoparticles surface modified with a zwitterionic pentapeptide designed from Bax-inhibiting peptide, or Ku70, to enhance cellular uptake and a linearized expression vector to induce BDNF in rat-derived mesenchymal cells. This demonstrated higher transfection efficiency than more complicated viral gene technology that we're used to, and around 80% transfection in a single administration was exhibited, as you can see by the expression of this BDNF-M-cherry fusion. as a testament to the in vivo studies, there has been some promising cases. For instance, transfected human fetal tissue derived neural stem cells with neurogenin 2 loaded polyamino ester based nanoparticles produced a large number of neuronal cells compared to non-transfected cells. Delivery of these cells with a hyaluronic acid hydrogel promoted vascular formation at the lesion site at 4 weeks following injection in a controlled cortical injury model. As you can see, as I talked about before, this combinatorial practice with materials, different types of payload is really the true path forward. As the lesion size alone can tell it all, but I'm really excited about going in this direction for future research. Thank you so much for your time, and I really hope that I've been able to convince you that nanoparticles have a lot of potential to address a lot of the needs seen in failed phase 2 and 3 clinical trials. Research is in the nascent field, but if you look at some of the clinical trials, you can see that if we can just increase this targeting efficiency, we can move forward towards finding a treatment. And I hope that these future studies can, look at more combinatorial therapies, consider the practicality of storage and formulation, and really I think there needs to be a lot of improvement to the injury model design, so that's something that I think would be a great field of research to explore. Okay, if you can bear with me for one more minute, I'd like to discuss one of my projects that involves nanotechnology, but in a little bit of a different light. So here I'm taking some control cortically impacted mice that I've performed the surgery. I wait a day, and this is the peak time for edema for mice, and I resected the swollen tissue, breaking down to single cells, and then added a deuterated probe, nanodots, and some phase change membrane, re-annealed, and then put them in an Enzo 640 library that already has FDA-approved drugs. It's quite random. And then I'm monitoring the efflux and influx of water, and after I've isolated those top candidates that did not allow a lot of water to enter the cell, I've said, okay, these are my top candidates, let's test them in vivo, hopefully to streamline some type of therapeutic, right, because it's already approved. So sorry for the secrecy, but I haven't published this results yet, but I've taken my most relevant drugs, meaning those that can cross the blood-brain barrier, and I've tested them 2 doses in a 24-hour time span. And from those that seem most significant, I'm now running a dose response assay. I actually have isolated my top candidate already. I did some dose response studies and think I've honed in on the right one. Let's hope. But the next thing to do is to run some behavioral assessments and sensory motor assessments, and then after that take my top candidate and submit that for some spatial sequencing. Right now I'm looking at 6 hours, 24 hours, and 48 hours post TBI, but incorporating that 24-hour drug data can really help identify some cellular targets and also some molecular targets. Because my data is bihemispheric right now, I can actually see where is this differential expression occurring. It might not be homogeneous, right? There could be some places that certain targets are upregulated or downregulated. And so my lab actually develops its own gene editing material. So I'm hoping that once I find a targetable pathway, then I can try to run some experiments in that direction. So thank you again for this opportunity to discuss my work, to be present in this conference. It's been a pleasure to learn from so many of you, just in life, reading your papers. It's really such a privilege, and thank you for taking this chance on me, a PhD student. So please, let's link. This is a QR code to my LinkedIn. I'm also looking for postdoctoral opportunities, so if you have any advice, please let me know. I'm willing to go wherever in the world. And thank you so much for your time. Thank you. This is my lab. This is the Joe lab here at Yale, and a professor that has been teaching me how to code because I am not a coder, and now I'm doing spatial transcriptomics. So thank you.